Hello everyone, this is Clement Chilambe from Clement Chilambe Tutorials, and I would love to welcome you to today's session, which is our tutorial 1B, which is just a continuation from the MCQs that we did in our first tutorial under the topic laboratory equipment and glassware. And this topic is coming from analytical techniques and laboratory methods. Okay, and we're focusing on short answer questions only. Why? Because we already did our MCQs. So the MCQs that we did in our previous tutorial, if you've not watched that tutorial, please go back and watch that so that you build on the knowledge with this second tutorial of ours. Okay, this tutorial, or these questions that are in this tutorial are designed to help you understand the content that you're learning and the content that you're reading in analytical techniques and laboratory methods, in molecular biology, and also other sections or departments of the biomedical sciences. This is fundamentals to biomedical sciences, by the way. So enjoy and ensure that you test your mind. Make sure that you also try to guess the answer, or to write down the answer, or to just tell the answer before we reveal the right answer. All right. Okay. So let's start with our first question. What is the primary advantage of using borosilicate glass over soda lime glass for laboratory glassware? Okay, what do you think this answer is on question number 1? Why should we use? What is the primary advantage of using borosilicate glass over soda lime glass? Okay, so the answer is here. So borosilicate glass has excellent resistance to thermal shock and chemical corrosion. Okay, so making it suitable for high temperature applications and use with harsh reagents, whereas soda lime glass is not. Okay, so this is the correct answer here. And when you are writing a test and you've been given this kind of a short answer question, make sure that you answer the way they are stipulated here. Can you see the answer here is actually correlating with the question that is above there? Just straight to the point, be accurate, be precise when you're answering questions, right? So let's go to the second question. Name 3 primary classes of biosafety cabinets. Okay, what does your lecture slide say? What does your book say about the 3 primary classes of biosafety cabinets that are used in diagnostic, molecular biology, or other departments of biomedical sciences? Okay, we have 3: class 1, class 2, and class 3 biosafety cabinet. Again, if you want to revise the comprehensive explanation among the 3 classes of biosafety cabinets that we have, please go back to the previous tutorial where we have analyzed each MCQ and given the proper explanation. Okay, we go to our second question, which our third question. So our third question, what is the key function difference between volumetric pipette and a graduated pipette? So make sure that you understand the question. That's one thing that I also wanted to comment. When you are answering questions, don't rush because you know the answer. Don't rush because you're just from revising your textbook or your notes about an hour ago. Ensure that you understand the question first before you proceed to answer. So the question here is, what is the key functional difference between a volumetric pipette and a graduated pipette? All right. So a volumetric pipette is designed to deliver a single specific volume, while a graduated pipette has markings to deliver a range of different volumes. Very, very accurate and very direct answer that one would wish to give during an exam or a test, right? Question number 4. In flow cytometry, what cellular property is measured by forward scatter? Okay, what cellular property is measured by forward scatter? So the side scatter, forward scatter, but specifically forward scatter, what we have. So forward scatter, we measure the cellular property that we measure here is the size of the cell or particle. What is the size of the cell or particle? We do not really measure the granularity or internal complexity. Okay, so granularity is for side scatter, but for forward scatter, please we do the size. We only deal with the increase, okay, of the cell or particle. Right. So let's proceed to question number 5 and 6. So 5, what is the primary purpose of an automatic tissue processor in a histopathology lab? So before we answer this question, I would love to announce something nice here. We will start doing our lectures apart from these tutorials that we're providing on this channel, and we'll be discussing the key pathology laboratory equipment, right? So from parasitology, from molecular biology, from blood bank or transfusion medicine to hematology, including histopathology. So we'll be discussing the principles of the pieces of equipment that we find there, because actually this is analytical techniques and lab methods. So we need to know and discuss the principles of those as well. Right. But this topic is only identifying laboratory glassware and equipment, right? Not really into the what is the principle or whatsoever. We'll discuss that with the corresponding the downstream topics that we have. Now, what's the primary purpose of an automatic tissue processor in a histopathology lab? So to automatically prepare tissue samples for testing by fixing, dehydrating, clearing, and infiltrating them with paraffin. That's the answer. That's the primary purpose of an automatic tissue processor. Number 6, which part of a microscope is used to make the fine adjustments to the focus after using the coarse adjusting knob? And I would love to mention that microscope is also an indispensable tool in diagnostic laboratory, because yeah, it helps us deliver the diagnostic function of the lab. That's why we find it in many of the laboratories. In histopathology, we have the microscope. In parasitology, we need to have the microscope. In hematology, even in blood bank, the issues of cross-matching, and also even microbiology, right? We need to have the microscope. It's a very, very important diagnostic feature. You need to know the parts of the microscope and their functions. So this question is just designed to ensure that you get some hints about some parts of the microscope and what they do. You need to think critically as well. Yeah, learn some good skills. Of course, we'll discuss most of the content about the microscope in this corresponding feature. Now, which part of the microscope is used to make fine adjustments to the focus after using the coarse adjustment knob? All right, which one is it? Which one do you think is it? The answer is the fine adjustment knob. Okay, the fine adjustment knob is the answer. Okay, question number 7 and 8. What is the defining feature of a class 3 biosafety cabinet? Remember, biosafety cabinet, class 3 biosafety cabinet from the previous tutorial. So what is the feature here? So the class 3 biosafety cabinet is a gas-tight enclosure, and personnel work through attached gloves, providing the highest level of containment. Remember, it is in this class 3 biosafety cabinet where the viruses such as the Ebola virus and SARS-CoV-2 viruses, the high-level viruses which can easily spread, are actually contained, right? They are discussed here. When we talk of the class 1 and class 2 biosafety cabinets, you know, in class 1 there's no product protection, but class 3 has got everything, right? Yeah, so you need to know this as well. What's the name of the laboratory instrument used to cut very thin sections of tissue for microscopic examination? Yeah, it's an indispensable tool in histopathology, and this is none other than a microtome. Yeah, in the previous lecture, or tutorial, we did discuss the rotary microtome and the cryostat, the difference between the two, and that was a very nice MCQ. But yeah, the only important thing here is knowing that a microtome is a tool or a piece of equipment that is found in the histopathology lab that is used to cut very thin sections of tissue for microscopic examination. Right. Question number 9, what is the function of the ejector button on a micropipette? Okay, you remember micropipette, we use small volumes, isn't it? We measure small volumes, and usually this is in, not usually, it's actually in microliters, right? So the function of the ejector button is to discuss the disposable pipette tip. Please follow through some of the content that we have under this section, and make sure that you also pay attention to when we are discussing this laboratory equipment and glassware, because there are actually other diagrams that really shows the clear picture of a micropipette as a laboratory equipment, right? So what is important right now is just to know that the function of the ejector button is to discard the disposable pipette tip. And what is the scientific term for the smallest distance at which 2 points can be distinguished as separate under a microscope? The answer is resolution. Sorry, I revealed this fast, right? I revealed this fast, but the answer is resolution. It's a scientific term for the smallest distance at which 2 points can be distinguished as separate under a microscope. And you need to know the difference between magnification, resolution, and some of the common terms that are used when it comes to microscope operation. Right. Number 11, what is the main purpose of a spectrophotometer? You know, this is spectroscopy technique, right? We're going to discuss this in the next tutorial. So the main purpose of a spectrophotometer is to measure the intensity of light absorbed by a sample, which can be used to determine the concentration of a substance. Yeah, that's the main purpose of a spectrophotometer. Yeah, this is where, under this topic, is where you find the Beer-Lambert's law, and we're going to discuss a lot of application of some of the principle, and also the application of spectroscopy as an analytical technique and laboratory method. Yeah. Which type of glassware is specifically designed to contain a single very precise volume? That's number 12. Remember, you need to pause this video and try to answer the question according to what you've read and what you've watched in other videos. But yeah, this tutorial is designed to ensure that you retain the knowledge, you practice, and you apply the knowledge that you have, isn't it? Now, let's answer question number 12. Which type of glassware is specifically designed to contain a single very precise volume? Which one is it? A volumetric flask. So if you did say a volumetric flask, then you were right. It is a glassware that designed to contain a single very precise volume. 13. What is the key difference between serological pipette and a mole pipette? Okay, the answer is graduations on a serological pipette extend all the way to the tip, while a mole pipette's graduations do not. So I'm going to show you the difference. I think I'll start incorporating diagrams, yes, in this question, so that you may easily understand what we're talking about, and you can easily relate to from your books that you're using, and also the lecture slides. Number 14, what is the primary use of an incubator in a microbiology lab? The use of an incubator in a microbiology lab to maintain a constant optimal temperature for the growth of microorganisms or cell cultures. Okay, when we receive a sample in microbiology, we culture it if it requires culturing, and then what do? We, after culturing it on the plate of the choice, not the plate of your choice, but they are different media that we use in microbiology. Maconchi, chocolate agar. Yeah, so after culturing, what do? We incubate for 48 hours, right? So when we incubate for 48 hours, then we read the plate. So incubation is very important to maintain a constant optimal temperature for the growth of microorganisms or cell cultures. That is our answer here. In flow cytometry, what property of a cell is measured by side scatter? I think when we did discuss, I know we did discuss forward scatter. I think that was question number 2 or something. Yeah, but let's talk of side scatter, the internal granularity or complexity of the cell. Remember, for forward scatter, we did talk about what? The size of a cell or particle. That's the property that it measures. But for side scatter, it's the internal granularity or complexity of the cell. 16. Name one key safety procedure for working inside a biosafety cabinet. Name one key safety feature or procedure for working inside a biosafety cabinet. Always work with the cabinet sash at the proper height to maintain containment. And then I had to include some of the answers here, which you can also put, which are also acceptable. Sterilize the work surface before and after use, and wear appropriate PPE, and also decontaminate regularly. Okay. Personal protective equipment. Make sure that you wear appropriate. Right. What's the function of the condenser on a compound microscope? And what's the primary purpose of an autoclave? Okay, so what's the function of the condenser on a microscope, compound microscope, is our 17th short answer question. The answer is to focus and concentrate light onto the specimen on the stage. Without question, clear answer. It's a short answer question. You don't have to explain. You're actually going to confuse us, but make sure you write these kind of answers as you're writing your exams or your tests. What's the primary purpose of an autoclave? Really, what is the primary purpose of an autoclave? Primary purpose of an autoclave is make sure that you sterilize equipment and media using high pressure saturated steam. So to sterilize equipment and media using high pressure saturated steam. This one you must know. Autoclave. Which part of a micropipette is used to draw up and dispense liquid? Which part of a micropipette is used to draw up and dispense liquid? Simple. The what? The plunger is the part of a micropipette which is used to draw up and dispense liquid. What is the main application of a polymerase chain reaction machine? What's the main application? The main application, so there are various applications, right? But the main one that we're looking for is that this PCR, polymerase chain reaction, found in microbiology, not microbiology, but molecular biology laboratory, is to ensure that it amplifies specific segments of DNA, making millions of copies of a particular DNA sequence, right? And this is known as DNA amplification. So amplification method. Remember, in a molecular biology lab, in an advanced one, you can start with the DNA extraction. This is where we actually isolate nucleic acids, right? By the procedure that we actually be given. I'm going to explain that later. And then we have the plating method, and then we have the amplification, which is by using the PCR. We're going to discuss all these under this procedure. This is actually the end of the short answer questions that we had today, right? So as usual, this is Clement Chambe Tutorials. You subscribe, you like, you comment, and you share as well. Thank you so much. Until next time.